CBSE Class 12 Practical Skills in Biology. Experiment number 8. Deoxyribonucleic acid, that is DNA. 8.1. To isolate or extract DNA from available plant material like onion, green pea seed, spinach, kiwi, strawberry, and etc. Aim: to isolate or extract DNA from available plant material like onion, green pea seeds, spinach, kiwi, strawberries, and etc. Materials required: 95% ethanol, that is 2 ml, thermometer that will measure 60 degrees Celsius such as candy thermometer, 250 ml beaker, 1000 ml beaker in 2 numbers, funnel. Alternatively, a strainer may be used that will fit in a 1000 ml beaker. Test tube with cap, watering pipettes. Alternatively, a pasteur pipette or medicine dropper may be used. Freezer, refrigerator, hot tap water bath at 55 to 60 degrees Celsius, ice water bath at 4 degrees Celsius, measuring spoons, light colored dishwashing liquid or shampoo, table salt either iodized or non-iodized of 1/4 tsp, distilled water 90 ml, spoon for mixing in 4 numbers, large onion, food processor, blender, a sharp knife for cutting the onion, timer or clock, filter paper, flat toothpicks in 2 numbers, and meat tenderizer that contain papain such as adults, glass rod, pasteur pipette that has been heated at the tip to form a hook or a similar device. Procedure: put the 95% ethanol in the freezer to chill it. You will use this later. Prepare 2 water baths, one at 55 to 60 degrees Celsius and other filled with ice cubes and water around 4 degrees Celsius. In the 250 ml beaker, make a solution consisting of 10 ml of liquid dishwashing detergent, shampoo, and 1/4 teaspoon of 1.5 grams of table salt. Add distilled water to the beaker to make a final volume of 100 ml. Dissolve the salt by stirring slowly to avoid foaming. This is solution A. Coarsely chop 1 large onion and put it into a 1000 ml beaker. For best results, do not chop the onion too finely. The size of the pieces should be like those used in making spaghetti. It is better to have a pieces too large than too small. Cover the chopped onion with 100 ml of solution A. The liquid detergent causes the cell membrane to break down and dissolves the lipids and proteins of the cell by disrupting the bonds that hold the cell membrane together. The detergent causes lipids and proteins to precipitate out of the solution. The table salt, that is NaCl, enables nucleic acid to precipitate out of the ethanol solution because it shields the negative phosphate end of DNA, causing the DNA strands to come closer and together. Put the 1000 ml beaker with the chopped onion and solution in a hot water bath at 55 to 60 degrees Celsius for 10 to 12 minutes. Continue to monitor temperature of the water bath and make adjustments as needed. Do not keep the mixture in the hot water bath for more than 15 minutes because DNA will begin to break down. Cool the mixture in an ice water bath at about 4 degrees Celsius for 5 minutes. During this time, press the chopped onion mixture against the side of the beaker with the back of the spoon. This step slows the breakdown of DNA. Filter the mixture through a filter paper placed in a funnel or strainer over a new 1000 ml beaker. While filterizing the onion mixture, try to keep the foam from getting into the filtrate. Put the whole setup in the refrigerator and let it filter overnight in case of slow. Dispense the onion solution into a test tube. The test tube should contain about 5 ml of solution or be about 1/3 full. For most uniform results among the test tube, stir the solution frequently when dispensing it into the tubes. There is not an advantage to dispensing more than 5 ml of solution into the test tube. The solution can be stored in a refrigerator for about a day before it is used in the following step. When the solution is removed from the refrigerator, it should be gently mixed before the test tubes are filled. Take the cold 95% ethanol out of the freezer and add it to the test tube to create an ethanol layer on top of about 1 centimeter. For best results, the ethanol should be as cold as possible. DNA is not soluble in ethanol. When ethanol is added to the mixture, all the compounds of the mixture except for DNA stay in solution while the DNA precipitates out into the ethanol layer. Let the solution stand for 2 to 3 minutes without disturbing it. It is important not to shake the test tube. Watch the white DNA precipitate out into the ethanol layer. When good results are obtained, there will be enough DNA to spool onto the glass rod. DNA has the appearance of white mucus. Observations: the white DNA precipitate out into the ethanol layer while all the components of the mixture except the DNA stay in solution. Result: the DNA has been isolated and it appears as transparent, slimy, and white mucus.